Chapter 5. No Reason to Stay in Science. The race to identify the lesion in the RAS gene became a grand event quite rare in basic research. MGM or Columbia Pictures might have turned it into a film if Hollywood wasn't so thoroughly convinced that scientists are a group of unphotogenic maladapts. The race had a defined goal: to find, for the first time in history, the molecular origin of human cancer. The rules for the race were clear. On the one hand, there was the oncogenic RAS gene, which initiated tumors. On the other, the healthy cellular RAS gene, which didn't. Somewhere along the DNA of the malignant RAS gene lurked a mutation that had turned it cancerous. All the scientists had to do was compare parts of the healthy gene with parts of the oncogene until they pinpointed the error. 3 contestants participated in the race. Enough for complexity and intrigue, but not enough to confuse the audience. There were unexpected twists, blighted hopes, and tragedy. And in the best tradition of Hollywood, the most thrilling element of the race was the astonishing prize at the end. But let's start a few steps back. When Bob Weinberg first thought about finding the mutation in the human bladder oncogene, he wasn't sure just how close his competitors were to being in a position to look for the same thing. At the time, nobody knew that Weinberg, Wigler, and Barbaseed were all working on identical oncogenes. Nobody knew that the oncogene from Earl Jensen's bladder carcinoma and the oncogene from the T24 bladder cancer cells were the same genes, and that the gene had been identified years earlier in the claws of the Harvey sarcoma rat tumor virus. In that brief, benighted era before the discovery of the consanguinity between human and retroviral oncogenes, Bob Weinberg felt confident that his lab was in control of the EJ gene. The group already had one important piece of information about the bladder oncogene mutation. It was fairly small. Right after Chiho Shi cloned the EJ gene, he looked for gross discrepancies between his oncogene and the normal proto-oncogene.